Hi, my name is Greg Hinkle. I'm the head of research informatics at Alnylam Pharmaceuticals. We're in Cambridge, Massachusetts, and we're going to talk today about a partnership between Alnylam and Paradigm4. And our title is, the places you'll go with 1 billion regressions in an hour. Next slide. So I'm not sure if you're aware, but we're in the midst of the greatest genetics project that's ever been undertaken, and it's based in the UK, where 500,000 people have volunteered both their genotypic information as well as their phenotype. So we have access to microarray data, whole exome, and soon whole genomes from 500,000 people. They've also very graciously and very courageously given access to their phenotypic data. So their medical records, including longitudinal medical records, lab results, fairly extensive survey results, and more things like imaging. So very high content, very large numbers. And as the slide says here, it's longitudinal data. So we're actually be able to look back in time through the medical records, and information is actually updated in real time going forward, even to the degree, here we are doing all this because of COVID-19, where we're getting weekly updates on COVID-19 data from the 500,000 participants. So this challenge of sort of the scale of the data, the constant input of new data is a real challenge for a small company, relatively small company like Alnylam, where we have roughly about 120 scientists. Next slide, Alex. So we decided about a year ago that we wanted to become a genetics company. And so how do you do that when you're looking at information at this scale? And we looked at a number of companies, and then we landed with Paradigm4. They have experience with very large data sets in scalability. Our goal really was to establish a world-class genetics capability. So we've built this up in less than a year's time. It was a novel capability for us, and speed was really important because we're part of a consortium of about 8 companies that's paying for this exome sequencing, and we get, in return for that, sort of paying for all that sequencing information, we have a 1-year exclusivity to look at the data. So time is of the essence here. So really, in deciding how to go forward, the real drivers were a willingness and an ability to handle the constant updates, and also to understand that we're going to have tremendous scale of both storage and of compute. And so how do we do that in a cost managing way? So I think Alex will take it from there.
Yeah, thanks so much, Greg. I'm Alex from Paradigm4, and we, as a company, have been co-founded by Mike Stonebreaker out of MIT. And Mike has been really on the forefront of so many database advances over the years. And the objective of Paradigm4 was really to handle large-scale scientific data. And the data sets like the UK Biobank with the genomics and the phenotypes are a prime example of the kind of problem that Mike really had the foresight to anticipate, and we were built to solve problems at that scale and that complexity. When we look at scientific data, what we see is a lot of dimensionality. So whether we are looking at signals over time with a time dimension, or when we are looking at genomic data, we have the genomic axis as a dimension, the position on the chromosomes. When we're looking at images, images are basically a multi-dimensional map or a matrix or a 3-dimensional block of data. So that dimensionality is really what unifies all of the scientific data and doesn't really fit existing database paradigm, and that's what SciDB, our product, was really designed to handle. In the life sciences, our key use cases include things like the biobank. In addition to imaging, obviously, single-cell RNA-seq, lots of dimensionality there, being able to sequence every single cell across multiple genes. Digital biomarkers, when we're sort of attaching sensors to people and capturing data about their activity and heart rate and so on. And the biobank is a great example because the biobank actually captures all of that. In the biobank, we have the genomics, and some longitudinal information, and there's some actigraphy, and there's images. And all of that data, having all of that data in one platform really gives us a lot of scientific power to be able to correlate somebody's traits or presence or absence of a disease, or something like susceptibility to COVID, to their genetic makeup and find the causes of disease and identify treatments. So the platform really has several pieces. We run in the cloud, and we've had great success running in AWS. Our system, SciDB, is optimized for storing and handling and computing on array scientific data. And then on top of that, we find that scientists prefer Python and R. Those seem to be the two languages of choice that are used in the scientific community. And we often also provide various applications on top of that layer. So things like working specifically with biobank data, working specifically with single cell data. Those are apps provided by Paradigm4, so scientists can get up and running, and we can go from a raw data set in AWS to a GUI in just a couple of weeks of setup. So with Biobank, our offering includes some GUIs on the front end to deal with the phenotypic complexity. There's a cohort selector that lets people just browse thousands of phenotype fields that we have for different participants. Then we have the package to analyze the data and run large GWAS. And then we have another UI called PheGe that lets people visualize the output of GWAS and really understand which genes or which genetic changes might be associated with which diseases or conditions. So as our users use the system, they sort of traverse this arrow across our applications, and all of that is handled in one place. So we provide both the GUIs and the heavy compute, and then the heavy compute creates data that can then be visualized in the user interfaces as well. So since we like multidimensional arrays and matrices, this slide really visualizes the problem nicely, the problem of UK Biobank. What we have at the outset is that in the phenotypic data space, 500,000 people, like Greg said, and for 500,000 people, we have 16,000 plus various pieces of information, various fields in the UK Biobank. Everything from age, sex, disease diagnoses in the hospital, primary care diagnoses, questionnaire results, really a wealth of information. And at the same time, for those same 500,000 people, we have their genotypes sequenced. We have the genomic sequencing data in the same place. And what people often do with this information is they clean up the phenotypes, and maybe they look at genomic locations of high interest or high quality genomic data, and they try to associate which phenotypes correspond to which genetic changes. So for example, which genes are associated with having high cholesterol or having a heart attack or a risk of heart attack. And there's all sorts of interesting challenges really at every step of this pipeline. If you just want to ask the question of how many people had a heart attack in the UK Biobank, well, you could go by the diagnostic code that we get from the hospital data. You could go by self-reported questionnaire data, and you find that those two sets actually don't overlap perfectly because there's errors in each set. And so there's pipelines created just for the purposes of identifying what the phenotypes are and curating those carefully. There's a lot of code that just does that. And then, of course, as soon as you do that, new data comes in, right? And this information gets updated all the time by the biobank. And then the next thing you know, the genotypic data gets updated because we're going from the imputed data to the whole exome to the whole genome sequencing. And every time this picture gets more and more clear. And then the next thing you know, a new analysis tool comes out. So maybe instead of doing this in a tool called Blink, we're now using a newer tool called Sage, which gives us a more precise statistical model of what the association is. And so we really do revisit this diagram over and over again and retrace the steps. And at every time, the researchers get a clearer and clearer picture of how the biology is associated with the outcomes. So our goal was to give the users, the bioinformaticists, an API like this, where they can quickly connect to a system and quickly kick off a job saying, in terms that they understand, that perhaps we want to look at 2 ICD-10 diagnoses like high cholesterol and heart failure, and we want to look at those diagnoses in the subpopulation that's British, from the UK Biobank, and we want to look at these 2 particular genes and only selecting variants that we think are of high impact, so only selecting the genetic changes that we think will actually have the probability of causing the disease, and we want to do it using a tool called SAGE. By the way, when we finish this calculation, when we do the statistical model, we want to actually preserve it in the database into something we call an association set, so that then this result is shared with other users of the system. So this is exactly the API that we've built, and these are exactly the kind of invocations that scientists at Alnylam are using to calculate statistics and to figure out the connections between the genetics and the phenotypic data. When somebody runs a job like that, what we've been able to do using AWS and sort of integrating SciDB with AWS is if there's a large calculation that happens, we can automatically scale out in the cloud. So we can automatically launch hundreds of worker machines to process the data. And using tools like Amazon S3 and a little custom code on top of that, we can make sure to send data to these workers quickly and run these large compute-intensive jobs. To the user, it finishes in about 10 and 15 minutes. But what happens in the background is like 200 machines come up in Amazon cloud, process the job, and then 1 or 2 of them may fail, but that's okay because other machines can come up and take their place. And then the result is all gathered, collected, and loaded back into the SciDB system. So this is the kind of thing where working with Amazon has allowed us to deliver this capability. It's something we call burst mode, being able to run these large calculations on demand using elastic scale out. So Greg doesn't need to buy 200 servers and put them in a basement somewhere. Instead, we can just rent 200 servers from Amazon for like 10 minutes. And then when we're done, we give them back to Amazon and we're only charged for the 10 minutes of time. This is really sort of the advantage of cloud that's allowing us to make progress so rapidly. So just some numbers here, right? If you're running on, let's say, 100 phenotypes and 10 million variants, so 10 million locations where people have genetic changes in the genome across 337,000 individuals, right? That's not too big, right? So 10 million variants is not a lot as far as the genomic data goes, and 100 phenotypes, 100 diseases isn't a lot of different disease types. You could definitely go a lot more. So this is sort of a medium-sized job, and if we ran this on one machine, it would take a lot of time. But when we run it on 200 machines in Amazon using 8,400 CPU cores, right, we can get it done in an hour if it's a linear regression model. And if it's a more complicated logistic regression model, it can get done in less than a day. And that's really the benefit. The scientists can get their results quickly. And because we use AWS Spot instances, the cost is the total cost that we pay is a lot less than what you would have to pay if you were going reserved or maybe trying to set up the hardware yourself. So it's just a great benefit all around. And finally, when these results are ready, they can be loaded back into the GUIs for the scientists to look at them. So you can, after these big jobs are computed, we preserve the results, put them in database, and we have great capabilities to query them, and the scientist can go and look for something like high cholesterol and look at what variants or what genetic changes are associated with that disease, or look at a particular genomic location and see what diseases are associated with that particular locus. As we extend our kit of tools, we really like to integrate with what's available out there. So there's lots of these different genomics tools that can compute these association scores differently. There's lots of different additional data we can bring in. We can annotate our genomic data using various sources. And there's various ways that people like to use the system. So we have an R API, a Python API. We have a couple of user interfaces that we're continuously improving, and so on. So this kind of offering is something that's that we have, and hopefully it helps folks like Greg tackle these really complex problems. And so really excited to be here and tell you about it. And back to Greg.
Okay, so thanks, Alex, for that description. I can say, we're about 18 months into this project, and I can say it's been a great success from Alnylam's perspective. We really went from essentially zero capability in genetics to a world-class capability, and it's because we have this world-class data set. We have a particular technology that lets us turn off any gene, and so 2 years ago, if you're going to ask what happens if you turn off a gene, the first thing you do is go look in a mouse. Nowadays, the first place you look is in the human genetics database. We go and find if there's a person in one of these giant databases that doesn't have the gene. What does their health look like? Are they protected from some disease? Do they have any phenotypes that we would try to avoid if we were to target that gene? So genetics, in the drug industry, where it costs billions of dollars to run a trial to bring a new drug to market, genetics data is absolutely the best data. And so we've been really happy with Paradigm4. We've been really happy that our compute cost to access the data and to manipulate the data in a genetic means is accessible to a company of our scale. And I'm really, we couldn't have done this without, both Amazon and frankly with Paradigm4. So it's been a great success.
Yeah, thanks so much. It's also been great to work with you from our end. Really appreciate it.